coming, but just looking at virulence factors, the capsule or glycocalyx, Fimbriae, pili, toxins, that's just kind of like preliminary example of the types of characteristics associated with a particular organism that will make it more virulent. Okay, so having a capsule, that gives it protection, and then by the definition of the virulence is what allows the pathogen to be able to cause disease. So having a glycocalyx that's large helps to protect the organism, that makes it more virulent. Fimbriae and pili help it do what? Not so much, a little bit, but attach, right? Remember those characteristics like attachment and protection were the things. This is where this, comes back in, remembering what you studied in chapter 3 with the bacterial cell. It can make them move a little bit. Remember, it contributed to like gliding motility and twitching and such like that. But even having flagella and allowing it to be able to move, to be modal, to spread more. You see what happens when you're looking at a plate of Proteus or Alkaligenes, right? You see it like spreading all over the plate. So you can imagine in the right conditions, being very modal like that would allow it to spread more. If it can spread and grow despite the host defenses and then cause additional damage because it produces toxins, compounds that would allow it to invade or destroy surrounding tissue or cause an effect in a particular area of the body, all of that makes it more virulent. Okay, so the fact that it's a pathogen means it can cause disease. The pathogenicity is its ability. The virulence affects that ability. The more virulent it is, the more of these types of characteristics it has that enable it to grow despite the host defenses, to cause damage despite the host defenses. Okay, so we're taking like basically those types of terminologies that you might have been used to hearing, but we're adding like the exact definitions and hopefully an understanding of like how this then is going to tie into causing disease. Something like this, pathogenicity islands, these are areas in the genome of the bacteria that they find that have a lot of these characteristics. So this is something that is associated with many pathogens. Many pathogens have these same types of characteristics. That's why they call it a pathogenicity island. So it's a section of the chromosome that has, and also it says here too, or plasmid DNA, that has many of these same characteristics. So characteristics that would increase the virulence. They don't see these pathogenicity islands in non-pathogenic organisms. And they also can be spread. So it's just a characteristic grouping of genes, basically, that contribute to virulence. Okay, so when you look at this, factors impacting the outcome of this relationship, you have the number of organisms present, the degree of virulence of that organism, so how pathogenic it is, and the host defenses, or the type of resistance somebody has. These things are all going to be related in a certain way. And before I go on to the next slide, if you just think of this ahead of time, you can imagine that if you have a large dose of an organism that has a large amount of virulence, together that's going to make really a big problem for the host defenses, right? So if you have a large dose, so the person's exposed to a lot of those organisms and they're highly virulent, you're pretty much certain the person is going to end up getting that particular infection. But if you look at something like, okay, it's virulent, but there's only really a small number of organisms, maybe only a few that somebody was exposed to, and the person has a lot of resistance. So they're very healthy, they're tip-top shape, they don't have any kind of immune disorders, then maybe they're not going to end up getting sick, even though it was a virulent organism. So there's a relationship there. Does that make sense, looking at it? And the relationship is really something like this. Whether somebody's going to get the infection or infectious disease really is the number of organisms times the virulence divided by the host resistance. That's the relationship. That's not like you're filling in numbers for them, but it does make sense, this type of relationship, because if you had a lot of organisms, but it was not a very virulent organism, then this top number would theoretically be much smaller. And if somebody had a lot of resistance, that would be a bigger number. It would make it much less likely that somebody would get the infection. Okay, so you can see how all of these really can relate to each other. If you had somebody who was really immune compromised, so a low resistance, but they were exposed to a lot of organisms with a high virulence, then you know that somebody is going to get an infection in those cases. So that's how those terms are kind of related to each other. And of course, the number of organisms can vary because you do have an infective dose. So that's going to be like the minimum number, basically. And usually when they come up with infective doses, they come up with something like the infective dose is the minimum amount that would get, 50% of the people sick. That's how they kind of come up with infective doses. Okay, and so we do know that that's going to really be influenced by how resistant a person is. And even the resistance that people have, even at their most resistant state, like they are their healthiest state, you know that there are differences between person to person and how they respond to infection. There are some people, like I mentioned, that student that I had who ended up with H1N1 and then the pseudomembranous colitis from Clostridium difficile, then, testing positive for tuberculosis because she was being treated by a nurse that turned out to have a positive TB. She was a person who always said that she always got everything. Whenever something was going around, she always got it. She seemed really normally healthy, young, no reason she didn't have any type of infectious disease or autoimmune disorder or anything that she was aware of. But she was just always the person who got everything. She wasn't sickly because she would always recover from everything, but you know those kinds of people. And then there are those people that swear they never get anything. Right? I know I told some of you, I don't know if I told the whole class, did I tell you about the student I had whose husband ate the dead fish? Just because I had a student whose husband was always complaining that she was always coming home and telling her all the, oh, you could get this and you could get that. And so to prove her wrong, because he was always saying, oh, I always ate like that and I never got sick, and he was from Mexico too and he never got sick and whatever. He ate a dead fish off the bank of the Kern River just to prove her that he wouldn't get sick. And he didn't get sick. So, good for him, I mean. I don't know if their relationship was ever the same again after that, but it's who would do that? But, he didn't get sick. But, I mean, the Kern River, really, and you never eat raw freshwater fish anyway, as a rule. But a dead one out of the Kern River, especially not good. So they do know that there really are individual differences in people's immune system that maybe at some point they'll know more about it and they would be able to explain why that particular student was always getting everything or why there are those people that just never get anything. But as of now, there is just really that range. if we look at the infectious disease process itself and look at basically kind of what happens when somebody, has the infection, what those different stages are, when they would be considered contagious and such, that's kind of what this next little batch of terminology is related. Because again, it's those things that you kind of take for granted, like, well, I know what that is, and I know, like, when you get symptoms, and I know, like, you have an incubation time, but it's just that time to think this is all, like, associated with the disease cycle and epidemiology. So, like, really looking at how things are spread, when somebody is contagious. There are all those rules, like real rules, not the, oh, you're not contagious after 24 hours on an antibiotic. See, I should put something like that on the exam, right? I should respond to that.
That's not true.
Why? Haven't you heard that before, though? Pardon?
Yes. Don't they tell you that at the doctors? They usually say,
Yeah.
you can go back to school after 24 hours.
Right.
I just went to school today.
Yeah, it all sounded good, right? Didn't you
Yeah.
just have this sense of like, oh, after 24 hours, okay, I'm counting down the clock. Oh, it's been 24 hours now, I'm not contagious anymore. Or now my child is not contagious anymore. We never talked about this in here? Oh, see, I could ask that on the test, right? Because, like, tell me what you think about that.
I thought it just killed probably whatever, like, the most viral ones that could get somebody else sick immediately.
What does anybody else think about that? Did it give you a sense of security? And that was, of course, after you were actually diagnosed with a bacterial infection, right? I mean, we didn't spend a lot of time talking about antibiotics, but antibiotics really for bacteria are going to be effective against bacteria, not against viruses, right? Or anything else. So when you got that antibiotic, and they never confirmed that you had a bacterial infection, I mean, they know the majority of times you don't have a bacterial infection, you have a viral infection. So then how did the antibiotic make you not contagious after 24 hours? I'm sorry, I'm like ruining everything for everybody, right? Honestly, like when my kids were little, I was more worried about my kids being around the kids on antibiotics. Because I would be more worried that my kid was going to pick up an antibiotic-resistant organism than pick up virus,
Did your kids go to daycare?
and preschool.
Because that's like, my kid gets sick all the time. They just give you an antibiotic and tell you, take that. Scary.
So it's like now you're kind of wondering like, what if those doctors know that you don't know? Do they know like something happens in your body after 24 hours and boom, those bacteria that might like get out, can't get out anymore or something? Like they're on they have their own little biological clock, oh, we give up after 24 hours. Like obviously that comes from nowhere. That's like a total one of those, I want to call it like old wives' tales, because it's not really coming from anything. There isn't any like, find me some kind of scientific study that even shows something like that. Not to mention that, of course, yes.
I never heard that, but I heard that you're not infectious after your fever was on. Is that the same thing?
It is the same thing, because like, again, what kind of research has ever been shown that when a fever is gone, you're not infectious?
You would think your doctor would have.
I mean, it would depend on like what you had, because you're infectious a lot of times without ever even having a fever, right? Like lots of times, like you get a virus and you never have a fever. You get a cold virus and you don't have a fever. Are you not infectious then?
I mean, I never agreed with that.
But it's just like why do they just give out like information like that? I mean, honestly, I don't know where it comes from, but you had your hand up. You were just scratching your head, right? You had a question. Why?
No, well, like, I know like virus, like, when you have a virus, you know, infections and stuff, and all, does it help at all if you have a bacteria infection?
Yeah. Antibiotics?
Yeah. I mean, like, as far as like, knocking down like common infections, you know, that.
Yeah, I mean, you'd have to think of like what people actually have that are bacterial infections though. Like when you get a respiratory infection, it's not very common at all for it to be a bacteria. And even when people have things like bronchitis, the studies actually show that antibiotics don't work very well against bronchitis at all. You know, even pneumonia. Because really it's like your body kind of has to recover on its own. So they're not very effective. So then that means like the majority of times somebody has a respiratory infection, antibiotics are probably not going to help them very much. And the majority of ear infections, the studies have shown that the majority of ear infections are viruses. And the studies have confirmed that whether kids are put on an antibiotic or not, they get well in the exact same number of days. So like, and I mean, that's even gotten all the way out into like over-the-counter literature. It still might make you afraid as a parent to deny an antibiotic if the doctor says that your child needs an antibiotic and they're like suffering with an ear infection. You know? I mean, people like, it's scary, right, to like make that decision. You do need the doctor to tell you whether or not they think that they really need one. But the doctors were just more likely to give out treatment because consumers are unhappy to pay a bunch of money to go see somebody and have them say, oh, you don't need anything. Remember, it was like the, oh, take 2 aspirin and call me in the morning thing. Anyway, it depends on whatever the infection is. And really, there aren't that many bacterial infections that are that contagious to people. You know, unless it was like you had some kind of skin infection that's going to be contagious to somebody. Yet on the other hand, you see kids all the time with herpes infections and they don't do anything about those, and those are totally contagious. And if you ever say to a child like, oh, you have a cold sore or something, everybody like immediately puts their hand on it. You're like, don't touch it. Because like it's completely contagious, right? So they could just be passing it on to other kids, right? Like most people don't even really realize that herpes are contagious, right? That always used to drive me crazy at the schools. Anyway, yeah, so it really depends. It depends on where you are in the stage, and like that whole thing of even being told about the infection, when you really look at it, even before somebody has a fever, aren't they sometimes known to be contagious depending on what it is that they have? It's just like, oh shoot, well I came down with these particular symptoms, but like I didn't get the fever until tonight, but I was just around everybody all day today, and everybody knows that, oh yeah, like they were contagious before they got the fever. So it's not that easy. There's nothing that's like perfect. When an organism is growing in your body, nobody is just like, oh yeah, at this today, yesterday they were contagious, today they're not. Like nobody has like a hard and fast rule about that. It's just that the doctors sometimes say, like I know having a discussion like this with my kid's pediatrician, they're just like, if you kept your kids home every time they were contagious or sick, they would never be going to school. Because they just like, that's what happens when they're little kids. They just like keep getting things over and over and over. So maybe like we just don't have to worry about it that much because they're gonna pick it up And then parents that have little kids, they get sick all the time too. And then there's a point where like everybody stops getting sick so often, because then you build up all these antibodies, and your kids like maybe have, built up all their antibodies, so they're not getting as sick. And then it starts again, I guess, in college, because then they start, getting together in like dorms and stuff. So then you start having things going around just because it's close proximities like that. But the things that usually go around are usually viral things. They're not usually bacterial things. Not usually. They can be like the meningitis. But so looking at these things, I think all these things you know already. Let's look at just like the course. So if you look at the incubation period, that's the period, you know what that is. That's when somebody has been exposed to, but they don't have signs and symptoms. The prodromal stage, that's when you're getting the signs and symptoms. But at that time, it may not be obvious what somebody has. Maybe they're coming down with a fever, their throat is hurting, like they're having some kind of signs and symptoms, but it's not really obvious to know like what they have. Then they have the period of illness. That's when it's the worst, and it does have particular signs and symptoms. And then the convalescent period, they're over the worst. Now it's obvious they're getting better. They still have signs and symptoms, but those are declining. And during the convalescent period, are people contagious during this time? Okay, because like if they're still coughing a lot, or sneezing, or maybe I shouldn't say they're still coughing a lot, they're still coughing, or they're still sneezing. If they have a cold, are they, because like a cold is an easy one that everybody is familiar with. Are they contagious anymore? Because the worst of it is over? Yeah, they're still contagious. So really, like, I think most of what people think of going into this, like, it's not exactly, what you think that it would be. If you look at the disease cycle itself, I mean, this to me is kind of it's not like really presented exactly in an orderly way with this for the disease cycle. Usually with the disease cycle, you're thinking of the fact that you have a reservoir. A reservoir can also be the same thing as a carrier. Or in here, they're saying a source. Okay, so these two really, they're written separately, but they really are kind of oftentimes one and the same. And that might be referred to as a reservoir. So when you think about the disease cycle, basically you think in the simplest state that you have somebody that is carrying the infection. They might have the infection, they might just be a carrier of the infection. Somehow there has to be some kind of portal of exit, it would be called. So how does that particular organism get out of you? Then how does it get from you to another person? Then a portal of entry, how does it get then into that person? And then you're back to that, reservoir, which is whoever has the infection or whoever is the carrier of the infection. Sometimes this could be two different things, like a carrier. And here they're almost making the carrier look like the same thing as a transmission. So I think it's like, I don't like this at all. So like, don't try to study by this particular diagram. Okay, at least it lists everything, but it's kind of confusing. So you think of the reservoir, you think of portal of exit, you think of the transmission. A portal of entrance, and then you're back to that reservoir. So it has to leave, it has to get there, it has to get in, as simple as that. just like learning about the life cycle and the parasites, it's important to have some idea about like how the disease cycle works for each type of infection, because that's how you can work to control that particular infection. So we know that, you know, the fecal oral route is huge, right? That is just the way, like basically it's through the hands. So what is the portal of exit? Feces. What's the portal of entrance? The mouth. What's the transmission? It's just a person-to-person contact or person-to-phone lines. So therefore, if you're trying to control something, of course you would try to control it at, you know, the transmission, at the exit, at the entrance, and that all can be controlled by washing hands. So, you know, really that getting something in the mouth is going to be really the most important thing. If you look at something like a flu, like respiratory infections, then you have the portal of exit. It's going to be through the, you know, lungs, through the mouth, and such, like coughing, sneezing. So the respiratory tract. Transmission could just be in the air. You could just breathe it in. And then the portal of entry is also going to be then through the respiratory tract, because you're going to breathe it in. And so you know, like, it's not, I mean, you're starting to see this more here, but you can see it a lot, like if you go to the Asian countries, or people like wearing like respiratory masks. Like it's not uncommon to see like people in Japan, people in China, and people here even that will wear some kind of mask just to prevent them from like inhaling, like when people, you know, are having flus and stuff. When you look then at this kind of more specifically, like the source or the reservoir, so you can see here, like here they have it source, but they don't say really anything about reservoir. So this can be one and the same. So you see where is the pathogen contracted, location from which it's transmitted to the host, may be the same as the reservoir. Okay, in the reservoir, and I think, again, it's probably easiest for me to think that if you just look at the reservoir, that takes care of everything, and just realize that sometimes that's where the source is going to be. Unless it just happens to be, you know, something else, and then it's given to the reservoir. So I think it's just easiest to think of it as the reservoir. That can be that natural environment which the pathogen is normally found. So that could be anything like soil, water, humans, animals, insects, and of course number one is going to be humans. But it could be someplace else, like there's a lot, the reservoir could be soil. Because things like Clostridium tetani, that organism can be in the soil. So therefore, that would be the reservoir, and that's how you can see that. That's when it really looks different, like the source and the reservoir. The reservoir usually is going to be whoever has the infection. Okay, so I'm not going to try to make some complicated, confusing, true, false, multiple choice about this because again, they overlap and it really depends on individual infections. So when a person actually has, so like if we look at a human and the person actually has the infection, okay, they're referred to as the carrier. Okay, so now it's like source, reservoir, and carrier are all like essentially in normal human infectious disease. They're essentially going to be the same, okay, unless it just happened to be from some other particular reservoir that got into that particular person. So if you look at that in that way, the types of carriers, you can have the active carrier, that's the person who has that overt disease, a convalescent carrier, which has recovered but continues to harbor a large number of pathogens. Oh, imagine that, right? So that just completely is like, where does some of the information that gets to you come from? A healthy carrier actually has the pathogen, but they're not ill. Okay, so that could cover somebody like the typhoid Mary situation, somebody that actually carries the organism, but they are not sick with the organism. And they really find that there are a lot of people in a population that carry pathogens without being sick. So if they looked at stool specimens of a lot of people, they would find that a lot of people are carriers of certain pathogenic fecal organisms. Maybe they had been sick, maybe they recovered, and they just maintained a certain flora. So they can look at fecal samples and they can find a certain percentage of people are carriers of salmonella. So people can get it that way. Other types of intestinal organisms too, for sure. So that's considered a healthy carrier. And an incubatory carrier is in that beginning time. They're incubating the pathogen in large numbers, but they haven't shown those particular signs. So that's one that you think of like probably most when you think about somebody having like say chicken pox or something. It's just like all of a sudden they come down with the actual chicken pox, they have the skin lesions, and then you think, oh, for the few days before that, they were contagious, only they just didn't know it. And then there's always like some like, well, when are they not contagious anymore? Are they not contagious when they all scabbed over? You know, it's not that cut and dry if you've ever taken care of anybody with chicken pox. It's like they're almost all scabbed over, but what about these few ones that look like they're not? So does that mean they're not contagious or they're contagious? You know, nothing is like completely cut and dry like that. This is all just like more examples of that, of just different organisms and what would be, you know, some of the reservoirs. The transmission then, that's pretty obvious what the transmission would be. Like things could be by direct or indirect methods. So that means like it could be, you know, direct person-to-person contact. It could be through a fomite. A fomite meaning like that pen that we were talking about at any place where you have to sign all of those purchases made on your card or sign into the doctor's office or whatever. You could have, you know, that pen that everybody's touched at the bank or anything like that. I mean, I don't think like a lot of people think how, if there's so many things that are the fecal oral route, why would you want to touch that pen that like 100 people that day already touched? And it's inside. And like, do they ever come and clean those pens? And then after that, and I know you would do this, Michelle, you would open up a piece of gum and take it out of the package and put it in your mouth, right?
Yeah.
That's the fecal oral route, or that's at least like, you know, the hand to mouth thing. So like there's a lot of ways, you know, in which like, when, like you don't like think, oh, I better wash my hands every time before you're gonna open a package of gum, or do you? Right? Because like just something like gum, can gum like pick up organisms off your hand? What, you said no? He just did that for you because he's your friend. And he doesn't want to make you feel bad. Also, they're saying it's not just enough just to make contact, like just to get the organism, you know, on your skin or whatever, but it actually has to come to whatever host tissue that it's going to infect. So there's more to it than just having an exposure. Okay, so that ability for the organism to get to where it needs to go in order to cause infection, that's going to affect its virulence as well. So if you have something that's invasive that can actually get into deeper tissues, that's going to make it more virulent. Modes of transmission then, like airborne, contact, vehicle, or vector-borne. So vehicle would be something like the fomite idea. Okay, so that you have some particular thing that has been contaminated that then is going to contaminate somebody else. Contact in this case is like person-to-person contact. And you know what vector-borne is, right? That's like through like a mosquito or a fly or flea or anything like that. So when you think back to kind of that original like terms that I drew on there, you have your reservoir. That reservoir can be the carrier, they can be the person that is sick or convalescing, or so they could be the source, but sometimes the source can be something else. Sometimes the source is, you know, through the soil or something like that. Then it has to have a way of getting out. So portal of exit, it has to have a transmission. It has to have a way to get back in. And then that's really the simple way of looking at the disease cycle. So airborne transmission, oh, that's such a good picture. Sneezing. I don't know if you guys were around when we did have the H1N1, if you were here. The college put a link on their website to that video about how to sneeze properly. You guys remember seeing anything like that? Because it really, the CDC was very concerned that year, their potential to have a lot of problems that year with the flu, because it was a bad flu, and there just was not very many of the people in the population that would have had any kind of antibodies, so there was really a big concern about that. And usually the way we sneeze is just something that, unfortunately, it's really not the best practice, because usually you sneeze into your hands, and then your hand is what's going to touch everything else. So even using a tissue or anything like that, then the tissue becomes like a fomite as well. And plus, does the tissue keep everything inside if you sneeze into a tissue? And then of course, our custom of shaking hands here, is something that's not really particularly good. That's why in a lot of other places, they don't do that. Okay, but that sneezing properly, you're supposed to sneeze into your elbow. Okay, have you guys ever seen anything like that? But it really takes a lot to undo a habit. And the video that went along with that, I don't know if it's still up, I could probably look for it. It's just kind of cleverly done, because they basically have all these people sneeze, and they rate them as far as how they do in the Olympics, and then the judges are talking about, if some of you have seen this one, but they talk about I give that person a 7 because, blah, 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 blah. But it's it was just entertaining. And also what they were trying to do is try to get this to be more socially acceptable, because it looks weird if somebody's sneezing into their clothes like that. So you would be maybe considered a polite person if you didn't sneeze in a polite way, right? So if you just sneezed into your clothes, that was kind of cute. But otherwise, you're trying to avoid all that, all those little droplets. There's a lot that's kind of understood about that when they look at droplets, not only the ones that spread, because you can see how far these would spread, but also how deep in your lungs they can go if you look at little droplets like that. I don't know if there's anything else more that I need to say about this contact transmission. About fomites. So you see here, vehicles and fomites is basically the same thing. Inanimate objects or objects involved in pathogen transmission, which again is what a fomite is. So you can use those terms interchangeably. Oh, that's even worse.
Oh.
What, you were eating gum?
No. I never eat gum.
Is it really true that it takes all those years to digest? No, that's not true. Let's see if there's anything else. we know about biological vectors and mechanical vectors when it comes to the vector stuff, right? You remember about that? You remember about the disgusting qualities that flies have that make them the perfect mechanical vector, and cockroaches. And you know what it means to be the biological vector. So here, this is what I mentioned about the infectious dose. So it's the number that will infect 50%. Okay, so that's kind of how they come up with that. The infectious dose 50. Okay, this kind of takes us into the information that is really just like another overlap of the stuff about virulence. Okay, so I think here you can just include these ideas because basically they're saying, okay, now you've had the exposure. So then what happens? Adherence and colonization. So remember the characteristics of virulence were the fact that it would be able to grow despite the host's defenses. And how is it able to do that? It has to adhere. And how did it adhere? The glycocalyx, the pili, the fimbriae, so all of those things. So this is really just an extension of the information about virulence. Okay, and it's here just because this is what would happen next in the disease cycle. So if you look at adherence and colonization, and this gives that more specific information. Okay, fimbriae, pili. And remember, I'm going to ask you to know the characteristics. So I'm going to ask you to be able to list me some of the characteristics that contribute to the virulence. Okay, so again, this is really just like information that you already had earlier in this chapter. Pili, fimbriae, adhesion molecules, the colonization, the fact that it could continue to colonize even despite host defenses. This gives you a list of some of the different particular compounds associated and structures associated with adherence. Adherence, if you remember back to chapter 3 when we were talking about it, this is important because if an organism is trying to establish itself, it has to be able to hang on to wherever it is. Otherwise, like if you're looking at the human body, things like mucus and blood and lymph and all of that will try to wash the organism away. It has to be able to hang on despite that. So those characteristics are important. The ability to invade. And invasion, again, can happen with those adherent types of structures, but also particular compounds that are produced, toxins and stuff that can allow it to invade tissue. Once it really gets into other areas too, then it's like past those natural immunities that you have in those areas. Like if it gets into deeper areas, it gets through the skin, if it gets in to other areas in the body, then it's really, again, that virulence that allows it to invade and get into those areas in which now you have less innate immunity. These are just some specific examples. Let's see, there's one other thing I want to get to, and then well, maybe we'll just go ahead. Are you guys like feeling like we've had enough information? Or you could even handle like another 10 minutes? Maybe we can do another 10 minutes? Because then we can just finish this off. Because basically what I want to get to is this next little part. Because this is really just talking about what we talked about earlier when we were talking about that relationship. So I'm not going to speak on this particular slide. But when you look at other things, biofilms, that would be another, virulence factor. Because if you have an organism that forms of biofilm, it's not going to be able to be affected as much by the host defenses. And now, this is the part that I want to get to, the endotoxins and exotoxins. So again, virulence, one of the factors that were mentioned was the production of toxins. If you look at the types of toxins that can be produced by the organisms, they tend to be divided up into two groups. There are others besides this, but two main groups, endotoxins and exotoxins. The endotoxins and exotoxins are more associated with gram-negative and gram-positive organisms also. And this is something when I was said before that there were certain types of symptoms that somebody might have that makes them more obviously infected by a gram-negative organism versus a gram-positive, and also then antibiotics come into play, because then if you have that idea as a clinician, you could be given an antibiotic that's going to be better for a gram-negative versus a gram-positive and such. And also how this all relates, the fact that the type of cell wall they have, and then the fact that we're originally only separated into these two groups just because of staining, but then later found out that not only was it just really something more important than the fact that they did stain in two groups, but the fact that it was associated with the type of cell wall, and now associated with the type of toxin they produce. So for the exam, and it is on here too, let's see. Oh, define, compare, and contrast exotoxins versus endotoxins. So I want you to know the particular characteristics. This table actually lists the characteristics pretty well, but they're also talked about on these following slides. So first, if you look at exotoxins. Exotoxins are mostly produced by gram-positive organisms. Okay, so that's this one right here. Most are gram-positive organisms. The fact that they're exotoxins, they're usually released into the surrounding. So exo, they're produced and released outside. If you look at something like exoenzymes produced by, say, something like bacillus, maybe that could remind you. Remember how bacillus was the genus that really produced a lot of the exoenzymes. So you looked at something like casein hydrolysis or starch hydrolysis, and those produced a lot of the enzymes, exoenzymes that are released into the surrounding area that cause a degradation of, casein or starch. That will help you remember that. So exo, they're released up to the outside. They're usually released while it's growing. Okay, and these are usually associated with gram-positive. It doesn't mean that a gram-negative organism doesn't ever produce an exotoxin or exoenzyme or such, but it's just more likely to be gram-positive. It says often travel from site of infection to other tissues or cells where they exert their effects. That's because exoenzymes are very, very specific. So if you look at, let's see, where is it? Here, specific host site exotoxins, neurotoxins, enterotoxins, cytotoxins. So exotoxins are usually very, very specific in their action. So usually specific and site specific. Okay, that's going to contrast with endotoxins that aren't. So we'll get to endotoxins in a minute, but already endotoxins are usually produced by gram-negative. Endotoxins are not specific. They're very general types of characteristics, and they're not produced and secreted on the outside, but they're inside, and they're released when the organism dies off. So exo and endotoxins are somewhat opposite of each other. When you see this one, soluble, most are heat labile. Okay, and one of the exceptions of that is that Staphylococcus aureus toxin that's associated with food poisoning. So, I don't know, like, I probably wouldn't memorize that one like I would these. Okay, so they're very, very specific. They're among the most lethal substances known. They are highly immunogenic. That means that they cause an antibody reaction. So something that's immunogenic means it's going to stimulate the immune system. Okay, immunogenic or antigenic, those terms are one and the same. Okay, immunogenic or antigenic. So they're highly immunogenic. They elicit a strong response. Because of that, and that's what this says too, they can produce antibodies. They can be chemically inactivated to form immunogenic toxoid. So it means that if you can get a strong reaction, then you can take those antibodies or those antitoxins and make something that can be used for treatment. Okay, so characteristics of the exotoxins, they're produced usually by gram-positive organisms. They're very, very specific in their action. They are strongly immunogenic. They can really, you can utilize that to produce antitoxins. They are very, dangerous. In fact, some of the most lethal substances. So something that would fit in here would be like Clostridium botulinum toxin. That would fit in there. That will help you maybe remember these types of characteristics. There's not a class waiting to come in here, is there? Yeah. I mean, I'm almost done, though. Endotoxins.
Did you say Clostridium botulinum?
Yeah. No. So with endotoxins, it's mostly gram-negatives. And you see here the lipopolysaccharide, right? Remember the LPS layer that a lot of you forgot on the diagram of the cell? See, all the stuff that you were learning from chapter 3 is coming back here again. Okay, so oftentimes that endotoxin is part of the cell wall. Okay. Also that lipid portion, the lipid A, that's attached to the LPS layer. The endotoxin is inside the cell. It's released when the cell dies, when it lyses open, even though some can be released when it's growing. So you can have a small amount released when it's growing. So primarily, though, it's released when the organism dies off. That's something good to know, because that way, if you have a massive infection with a gram-negative organism and then are treated with an antibiotic that is a bactericidal antibiotic, it's going to kill off those organisms, and what could happen? All those toxins released into your circulatory system. So if you knew this kind of information as a clinician, you wouldn't give somebody a bactericidal antibiotic. You would give them a bacteriostatic antibiotic to prevent organisms from growing, not necessarily kill those off. They will die off. But this is part of the reason why people can go into shock, with some of these types of infections, when all of that is released at one time. These tend to be heat stable. They are very toxic also, but they're weakly immunogenic. And they're generally similar despite the sources. Okay, so whereas the exotoxins were very specific, neurotoxin, enterotoxin, cytotoxin, these are generally causing things like fever, weakness, diarrhea, inflammation. The kind of general, somebody feels sick, but not necessarily those really specific things. So you can see where some of this information then is very helpful on treatment, because if you know that, then that gives the doctor additional information and how to treat somebody. If they have an endotoxin producing infection, they're not gonna wanna kill it off at one time, and they might recognize this by just these general things. They have a high fever, they don't have something that's really specific, versus the exotoxins. This gets into a little bit so more specific about that, which I don't think, there's none of these that you really have to memorize for understanding the difference between the two. So here, like resisting host defense, I think mostly we'll be talking about, I guess it's kind of worth mentioning this stuff now a little bit. I mean, this is basically kind of talking about what the organism can do to resist, defenses that are not some of the things that we already talked about before. But no, I don't think I'm going to talk about that. Yeah, I think we'll leave that here. We already know about exiting the host because we already talked about that. Those couple of slides are kind of the things that the bacteria can do to kind of fight off the immune system.